Hello everyone. Our topic is silencing the DUX4 gene, CRISPR epigenetic therapy for FSHD. To begin with, let's consider a simple question. What if your muscles slowly forgot how to move? For thousands of people living with a rare genetic disorder called facioscapulohumeral muscular dystrophy, this isn't imagination at all. Their muscles of the face, shoulders, and arms slowly weaken over time. This is our contents. So facioscapulohumeral muscular dystrophy, or FSHD, is a rare hereditary muscle disease. It is inherited in an autosomal dominant pattern and currently has no cure. FSHD affects about 87,000 people worldwide, roughly 1 in 15,000. The disease mainly weakens the muscles of the face, shoulders, and upper arms, leading to progressive difficulty with movement and daily activities. These diagrams show the typical pattern of muscle involvement and an example of the shoulder blade prominence seen in many patients.
In August 2025, a major breakthrough made global headlines. CRISPR biotechnologies treated the first ever patient with EPI-321, a new CRISPR-based epigenetic therapy for facioscapulohumeral muscular dystrophy, or FSHD. This marked the first time a gene silencing CRISPR therapy had ever entered a human trial. And what makes this technology so different is that it doesn't cut DNA at all. EPI-321 is built on a next-generation platform called CRISPR 3.0, unlike traditional CRISPR-Cas8 system that permanently adds GMO. CRISPR 3.0 is designed to fine-tune gene activity by turning genes on or off in a controlled, reversible way. At the center of this system is GEMS, the Gene Experience Modulation System. GEMS use a catalytically inactive Cas9 or dCas9 fused to special epigenetic proteins. Together, they can silence or activate genes without making a single cut in a genome. This is a safer, smarter way to control harmful genes, and it forms the scientific foundation of EPI-321. To understand why this therapy is so promising, we need to talk about one gene, DUX4. In healthy muscle, DUX4 is supposed to be completely turned off. It's likely wrapped in closed chromatin, heavily methylated, and totally silent. And it should stay that way for your entire life. But in FSHD, this silencing system breaks down. When DUX4 is accidentally switched on, even at very low levels, the consequences are severe. DUX4 produces a toxic protein that disrupts normal muscle function, triggers inflammation, and causes cells to undergo apoptosis. Over time, muscles weaken, waste away, and can repair properly. So the core problem in FSHD is not muscle weakness itself. The problem is loss of epigenetic control over DUX4. There are two major forms of FSHD, but both ultimately lead to the same issue. In FSHD1, the D4Z4 repeated error on chromosome 4 becomes abnormally short. This short repeat cannot maintain the normal closed chromatin state. As a result, the chromatin loosens and DUX4 becomes active. In FSHD2, the repeat length is normal, but mutations in epigenetic regulators, most commonly SMCHD1, prevent the gene from staying silenced. Even with the right DNA structure, the silencing machinery simply doesn't work. So whether it's a structural problem in FSHD1 or a regulatory problem in FSHD2, the outcome is identical. DUX4 turns on, poisons the muscle cell, and begins the cycle of degeneration. Once DUX4 is expressed in adult muscle cells, it activates a harmful genetic program that should only exist in very early embryos. This leads to oxidative stress, mitochondrial fusion, immune activation, and ultimately muscle cell death. This is why the disease process graduates over time, why different muscle groups weaken, and why there is currently no cure because none of the available treatments directly address DUX4. EPI-321 aims to fix this problem at the source. The therapy uses CRISPR 3.0 and GEMS platform to deliver dCas9-based epigenetic silence directly to the DUX4 promoter. Instead of cutting DNA, the system re-establishes the natural epigenetic marks, such as increased DNA methylation and tightly packed chromatin, that normally keep the DUX4 silent. In other words, EPI-321 attempts to restore the gene-off state that healthy muscle cells maintain throughout life. This approach is high precious because it only targets DUX4 locus, potentially safer because it doesn't modify the DNA sequence, reversible, aligning with the natural biology of gene regulation, and most importantly, disease modifying because it acts the true root cause. If successful, EPI-321 could represent the first therapy that meaningfully interrupts the molecular cascade that lead to muscle degeneration in FSHD.
There's a study conducted by Damon et al. In 2025, testing a new RNA interference strategy designed to shut down DUX4 in FSHD. They developed a small interfering RNA called DUO1 and attached it to a docosahexaenoic acid lipid so it could be delivered systematically. In FSHD patient-derived muscle cells, this siRNA strongly suppressed DUX4-regulated genes. To evaluate its effect in vivo, the team implanted human FSHD muscle cells into mice and treated them with lipid-conjugated siRNA by subcutaneous injection. The treatment achieves more than a 50% reduction in DUX4 downstream gene expression in the xenografted muscle. Although the study did not measure improvements in muscle strength, it clearly demonstrates that DUX4 can be silenced in vivo through a systematic RNA-based approach. This work provides important groundwork for developing future therapeutic strategies for FSHD. So to summarize how EPI-321 works, here are the key points of this treatment. EPI-321 is a new epigenetic CRISPR therapy designed to switch off the DUX4 gene. Unlike traditional CRISPR systems that cut DNA, this approach modulates gene expression without altering the genome, making it both precise and reversible, with a much lower risk of off-target effects. By silencing DUX4, the therapy interrupts the toxic cascade that leads to muscle degeneration. In August 2025, CRISPR Biotechnologies reached a major milestone by dosing the first patient with EP321. This is the first time a CRISPR-based epigenetic treatment has ever entered human trials. In phase 1, the main goal is to evaluate safety and tolerability, so the patient will be monitored closely to see how well EP321 can silence DUX4 and whether it improves muscle health. This first dosing is not only important for FSHD patients, but also a landmark moment for epigenetic CRISPR therapies, showing that gene silencing approaches can move safely from the lab into real human testing. To summarize, FSHD arises from aberrant activation of the DUX4 gene, leading to the toxic protein production and muscle degeneration. EPI-321 uses a targeted epigenetic CRISPR approach to silence DUX4, and the first human patient has now received this groundbreaking therapy, moving from theory to clinical reality. Looking ahead, researchers can continue to monitor patients for safety and efficacy as EPI-321 progresses through later clinical trials. This therapy has the potential to improve muscle function and slow disease progression. Future research can also enhance long-term durability for DUX4 silencing, and also design personalized therapy for FSHD1 and 2, such as chromatin target therapy for 1 and epigenetic enhancing therapy for 2. Finally, EPI-321 is the first, but it won't be the last. The significance of its success reaches far beyond FSHD itself, opening new therapeutic pathways for a wide range of epigenetic diseases. This is our reference list. And that's all. Thanks for your attention.